get started now. I'm Grant Haines. I'm a postdoc at Holar University. And I'm going to be talking about a really cool system we have of Arctic char that live in caves and a bunch of replicated populations and body condition in them and what it can tell us about evolution and ecology. I'm going to fly through this because I missed 2 minutes at the beginning. So if I run through anything that you're more interested in and I don't have really time to touch on, just flag me down afterwards. Also, everything in here is pretty tentative, so doesn't necessarily have the freshest, crispest data in all the new models. Yeah, just keep that in mind. Okay, so I'm going to be talking mainly about body condition. So this is a really important variable that's a little bit controversial for what it can actually tell us, but it's also quite easy and convenient to measure on live fish that we can release back into the environment. So it's basically just a ratio of length and weight based on predicted weight at a given length. It's often assumed to indicate health and fitness in individuals, but also health of populations or responses to particular disturbances. So what I'm interested in exploring in this system is body condition a good predictor of survival? What causes condition to change through time in a population? And what are the ecological and evolutionary consequences of body condition variation? So this is our system. It's around this large lake in these little caves that are formed by lava tubes, basically. So we have 20 populations that have very small population sizes, so a few dozen to a few hundred adults. We've been going back since 2012 and tagging them, pit tagging them so we can individually ID them, measuring them, photographing them for morphometrics, fin clipping them for genetic work, and weighing them. So this is what one of these little caves look like. So we just throw a bunch of minnow traps in there and catch some char and measure them. And this is what it looks like in April. That's also what it looked like for a bit of June this year. Yeah, growth and condition in these populations are super plastic. The fish can live over 10 years, but they rarely exceed 15 centimeters. So they're a bit unusual, and they can get these pulses of growth in the summertime especially, so they can grow like 2 or 3 times more in the summer compared to the winter. So just thinking about growth and condition, so because condition factor is a ratio of weight over predicted weight given length, if you take in mass through eating prey, you can devote that either to putting on weight without adding length or growing. So if you just use that biomass to put on weight, then your condition increases. But if you're devoting it to growth in length, then the condition might decrease if you're not sort of adding weight to keep up with that ratio. And so one of the things I'm interested here is the effect on survival. We want to show that like condition actually means something to this population and to these individuals. So I built a model to ask whether it's dependent on, or is survival dependent on length, using this mark recapture data we have from all these tagged fish. And if condition and length interact, is condition early in life going to have a strong influence on fitness, especially if most reproduction occurs later on in life, which we think it does. So yeah, there were a bunch of variables in this model that came out pretty significant. Condition directly wasn't one of them. So right here, Kn is not significant on its own, but it interacts with a bunch of other things. So it does interact with cave and also so population, and also the length, body length of the individuals. So here are 2 of the partial effects. So the left is the insignificant effect of length. It does increase slightly, but again, it's not usually repeatable. And that effect of length on survival is really interesting because that is a significant effect. So body condition has a positive effect on survival at sort of intermediate lengths and also really large lengths, but with a sort of weird little dip in between there. And it's negative, that effect is negative for the really young fish. I'm just going to skip that. So this is fork length on the x-axis and condition on the y-axis. Basically, the positive effects on survival are in the like gray, green, gray colors, and the negative effects are in the red. So you can really see that flip with the increase in age. There's a sort of threshold age where it becomes a positive effect after the really young stages, life stages, and like for the very old fish. So one of the ways we're getting at how the age is important basically is constructing pedigrees from the genetic data we've been collecting. So we can use those pedigrees to determine when in life fish are reproducing a lot. And it turns out that it's later in life. So that the plot on the bottom there is basically from this pedigree reconstruction model, it's updated priors of the age difference between parents and offspring in the 2 left columns. And then the 3 right columns are age differences between siblings and half-siblings. But you can see the greenish little boxes are when you're most likely to, the most probable age differences between parents and offspring, and it's like 8 and 10 years old. So basically the fish that are living the longest are reproducing the most in this population. Similar kind of story in this population, but more with the paternal, with the fathers, not so much with the mothers. But this is a slightly smaller population, so things are a bit noisier. And that's a relationship matrix, gonna skip that. So that's condition as a property of individuals and its effect on survival, but we can also think of condition as having an effect on populations, on behavior, activity and movement, intraspecific competition, reproductive investment, but also population dynamics and evolution. So the higher relative conditions, if they contributes to fitness and how active the individuals are, there can be really significant cohort effects that result from that. So if one size cohort is doing really well, it can kind of outcompete smaller or larger size cohorts for food and for reproductive success. So we can think about population level condition in 2 ways. You can think of mean population condition and also variation through years in the relationship between condition and length. So you might have 1 year where the big fish are doing really well compared to the small fish, and other years where the reverse happens. And we have that here for both of those here for all 20 of these populations that we've been tracking. So the left is the mean body condition, and this x-axis is ratio relative to predicted condition at a given year. So above 1 is like unusually high condition, above 1 or below 1 is unusually low. And the sort of slope that I mentioned before is on the right. Those effects don't look big, but the 0.02 there is 2% increase in condition per millimeter. So that would be like a monstrous effect if it were all the way up there. So it does fluctuate by season and by year, and some of these are evident like across multiple populations in the same years. This is autocorrelation, so it's these condition effects at the population level are also persist for over a year. And then I mentioned intraspecific competition before, so these individuals compete for food. The food does differ between the larger and smaller cohorts. So this is gut contents from fish. Along the x-axis is length, and you can see like the larger fish consume like way more adult flies. So from some guts we got over 1,000 adult flies. and there are lots of flies around. This is Cladocera. So this is a prey item that persists in the populations year-round. And we know that fluctuations, between-year fluctuations in the fly counts especially, have a huge ecological effect. So the lake that these populations are around translates from fly lake in Icelandic, basically. So they have these huge boom and bust cycles that influence diving duck recruitment and also fish recruitment in the nearby lake. So we think this is really important. So I've gone a bit over. So I'll just say thank you very much, and please see me if you have any questions.
Hello. Thank you for coming to my talk. My name is Anibal. I'm a PhD candidate in the University of Guelph. Today I'm going to be talking to you about one aspect of my thesis. So we know biodiversity is very important, and it has been recognized as such by the Global Biodiversity Framework, the Convention of Biological Diversity recently. Particularly, terrestrial ecosystems are very important, and the United Nations has recognized them as one of the Sustainable Development Goals. Now, I want to start, I said I'm going to talk to you about my PhD thesis. I'm going to start with my research question, just to put a framework for what I'm going to talk to you about today. So I'm working on the ecological drivers in community assemblages, in soil and leaf litter invertebrates, in ecological gradients. Particularly, I'm going to be talking about the temperature gradient today. So, how do we study usually leaf litter biodiversity? This includes the collection, the extraction of fauna. Here's a Winkler extractor, for those who don't know. Then the sorting of that fauna, and the imaging and identification. Now, traditional methods are rife with challenges. For example, if we gave a sample, a leaf litter fauna sample to 3 different researchers, we might get 3 different answers. And this would be very time-consuming. And also, these tend to exclude fauna that live inside plants, like gall-forming insects, and also leaf litter mining insects. So, what do we do about this? Now, a German group used store-bought tea bags and applied high-throughput sequencing to study the leaf litter, the diversity of arthropods from the plantations, the tea plantations that the tea came from, and they were able to reconstruct the arthropod communities from those plantations. Then they tried a very similar approach with herbarium leaves, and they were able to reconstruct like a time series of the variation of the arthropod communities according to the age of this leaf litter. Sorry, I keep saying that. The herbarium leaves. So I had an idea. What if we approach leaf litter with high-throughput sequencing? Because so far, leaf litter has not been approached with high-throughput sequencing. So I went to a woodlot in the campus of the University of Guelph, and here you have an aerial view that has a split between a forest and a field. The forest is a bit colder, and the field is a bit hotter. So I sampled leaf litter according to this sampling design. I took 5 samples in each environment, separated by 15 meters each. Now you might be wondering, where does this design come from? And this has been created by a call for standardization of the sampling methods for terrestrial biodiversity assessments. You can find that paper here in this barcode. And in each of these places I collected, in each of these spots I collected samples of leaf litter with quadrat of 25 by 25 centimeters. So, and then I went into the lab. So, my method includes drying and grinding the leaf litter. And here's the thing. The 2 methods that I just mentioned with the tea bags and the herbarium leaves, they used 2 different DNA extraction methods. It's unclear why, because they didn't compare them, they don't refer to each other, even though this is the same research group, and both of them worked. But still, to validate a pipeline, which is this, the main aim of this chapter, I used 3 different extraction methods: DNA-EC PowerSol Pro, the DNA-EC blood and tissue, both from Keragen, and Quick DNA plant and seed from Zymo. Then I amplified the CO1 gene, which is the standard for animal barcoding applications. And then I put that through high throughput sequencing in a MySeq platform. So my prediction is that the protocols will vary in their ability to recover this biodiversity. And I will consider, because of all of them come from the same sample that I split with these 3 different extraction methods, I will consider the winner the one that recovers the highest, the highest amount of different species or taxa, which is we refer to as richness. You know. Now, imagine the concept here of total biodiversity. Like I said, the protocols will vary in their ability to recover this invertebrate DNA. There will be some overlap, there will be differences in the amount of richness that they can recover. So what I found was that so the blood and tissue seems to work a bit better. As you can see, it recovered more samples from the field, higher diversity in the field, more samples overall. There seems to be more variation and more diversity in the field as opposed to the forest. And you can see by color that the field, all the methods vary in their ability to recover the diversity in each environment. Now, to see this a bit more clearly, here is an NMDS plot which explores the diversity. And here, by extraction type, again, we see that the blood and tissue recovers a broader range of diversity. If you remember this here, this is as close as it gets to a graphical representation of that concept of total biodiversity. And if we split that, I said I was working with a temperature gradient. So look at what happens, this beautiful split when I code the samples by environment, field and forest. What this is telling us is that the 2 environments barely have any species in common, even though they are adjacent. So this opens the several opportunities. For example, this method approximates a comprehensive survey of terrestrial fauna. If you see the literature, the assessments of fauna usually are taxa-driven. Somebody who works on ants will study the diversity of ants, just ants in that environment, or maybe a particular order of insects, and so on. But this is a very easy way to assess what we can hope is a comprehensive view of all terrestrial fauna. Now, this has other applications like screening leftover plant residue for pests, invasive species, or other species of interest. This can also be done in a variety of environments. I did this in a field and a forest, but this can be done in agricultural fields, in grasslands, and whatnot. It's also time efficient because this leaf litter can be collected right away, can be collected very quickly, then processed in the lab in a few days, and then you wait for the results, for the sequencing results for a few weeks. Give or take, that varies a lot. But it's you just waiting. It doesn't take your time to do, you can do something else. And also it can be cost effective, as depending on the way you sample and the way you sequence, it might be more, less expensive than traditional methods. And also it offers an out-of-season window into sampling because, for example, you can go in the winter and assess you dig in through the snow, if there is snow in winter where you're sampling, and you get the leaf litter, and you can peek into what was there the summer before. You're not constrained by time or by season. Now, no method is devoid of its challenges, of course. So this might not be appropriate for all environments. Some environments are not DNA friendly. For example, environments with high water content, like waterlogged environments, bogs, wetlands, marsh, and so on. It also depends on the existence of a reference library. When we get those sequences, we need a robust reference library to match them to. And in this case, I chose this woodlot in the University of Guelph's campus because there has been a lot of research done there already. And in fresh leaf litter, fauna might not have been left behind. Fauna might not have left DNA behind. Now, in conclusion, we were the first ones, to my knowledge, applying high-throughput sequencing to leaf litter. Check. Grinding and drying and grinding the leaf litter to assess arthropod communities works. Yay. And then we applied that successfully to differentiate two environments, two adjacent environments, and distinguish them in terms of their fauna. Check. So this is soon to be submitted for publication, so fingers crossed and wish me luck. I want to acknowledge the NSERC and Foods from Thought for funding the project and my student stipend, and for the last 3 years I've been the recipient of the Ontario Graduate Scholarship. Wish me luck for next year. And also the Arthur D. Latournelle Travel Grant and the American Society of Naturalists for allowing me, funding my stay here. And also want to acknowledge the great support from my co-advisors, Dr. Smith and Dr. Stanke, and my advisory committee, Dr. Jacobs, and their amazing groups, which have helped me a lot throughout this. And with this, I thank you very much. Questions, comments, suggestions, or you can reach me out later. Thank you very much. Here, we have time? Yes, go ahead. Oh, sorry. Go ahead.
Okay,
Somebody.
Just a quick question. We are doing something similar, but with rDNA,
Okay.
and we are comparing it with the baseline method, which is the Malastraps. So
Yes.
these one are like the super invasive, they catch everything.
Yes.
And we want to see the representation of samples that we get from DNA from the Malastraps.
Okay.
Do you know if on the leaf samples there is like a standardized protocol that gets you as much as possible to see how much of the species you're recovering with DNA from these traditional taxonomical methods?
That's one of the reasons I came up with this method, that all the traditional methods to address leaf litter diversity tend to be taxon specific. So you need to collect with different methods, different techniques and equipment to address as many taxa as you can, to even think that you are approximating a comprehensive assessment of that. And with this, you get everything right away. Yeah. It would be very interesting to do that, to recover as much as you can, to compare the two, and also to reinforce the reference library of what you're looking for. Yeah. But what I saw a review, and I can email you that if you contact me later, where they concluded that there's, with 7 different methods, you can kind of capture as many taxa as possible.
Okay, interesting.
Does that answer your question?
Yeah, perfect. Okay. Thank you.
Okay, in the interest of time, I'm done. Thank you very much.
All right. Sorry for the delay there. Hello, everybody, and welcome to my talk. My name is Zach Lange. I'm about to go into my 5th year of my PhD with the Frischkopf Lab at the University of Texas at Arlington. And today I'm going to be talking about kind of a summation of a lot of the work that I've been doing for my PhD for these last handful of years. So the focus of this talk is about the connection between evolutionary convergence or convergent evolution and community convergence. Basically, does one lead to the other? Are they related in some way? I think we're all pretty familiar with evolutionary convergence, basically when different species evolve similar adaptations to occupy a similar niche. It's pretty well documented across the tree of life, and I think probably this week we'll hear a lot about some different adaptive radiations where we see a lot of convergent evolution pretty widespread. Now, there have been a lot of studies that look at like convergent evolution and say that there's community convergence because there's a bunch of species that kind of match up in morphology or ecology, like in this example study in North America and Australia. What I'm talking about is a little bit different. Like, I would argue that this is an example of species for species matching, which is just like super widespread convergent evolution. What I'm interested in is seeing how this type of convergent evolution in these regional species pools could lead to similarities in communities that those species filter into. Now, kind of like the traditional way that people look at community level convergence is through species richness. And so this is kind of a classic study by Dolph Schluter back in the 80s, where he sampled species richness of finch communities across the world. So each of these different symbols represents a different continent, and you can see across these environmental gradients that species richness seems to follow this similar relative pattern. It's pretty cool, and so yeah, this is an example of community level convergence. So again, my focus is what is the relationship between widespread evolutionary convergence and this community level stuff. Now, there's a host of different metrics you can use to investigate evolutionary convergence or community convergence. For my talk today, I'm keeping it pretty simple. I'm looking at morphology and species richness, kind of the classic stuff. And the study system I'm using is direct developing frogs. So direct development is actually itself a broadly convergent reproductive strategy in frogs. It essentially liberates them from needing to be near bodies of water for their reproduction. These guys tend to be super abundant in the leaf litter and sometimes in other environments as well. And again, it's found throughout the phylogeny of frogs, so pretty cool. And I focused on 3 broad regions of the world from which to collect data. So I've got the Greater Antilles, minus Cuba, and then South America, specifically in Peru and Madagascar. In each of these locations, I've been doing work in 3 different landscape types or habitat types: the hot dry forest, warm wet forest, and cool wet forest. So these are different habitats that have been matched based on climatic similarity for the work. So that's kind of the broad overall study system that I'm working in. So real quick, let's get into evolutionary convergence. I have a lot of different hypotheses. I'm not going to bog this down too much with really going into them, but just going into this, I really wanted to identify what's the driver of morphological convergence or evolutionary convergence. And if we see similarities between one regional species pool and another, what does that mean? And so there's a host of potential explanations. Maybe if there's a bunch of other competing lineages of frogs, the direct developing ones are kind of constrained into a smaller range of morphologies and ecologies. But yeah, in order to get data for this, I measured a ton of museum specimens. I also collected a handful of my own and used kind of the traditional measures that we use for measuring frogs. I also got an undergrad from UTA, Dalton, who's now actually working on his master's to help measure a bunch of stuff in ImageJ, kind of the smaller structures. And from that, I decided, first and foremost, just to throw it all into a PCA, see if there's a bunch of cool convergence stuff going on. And what I kind of came up with was okay, depending on which PC axis you're focusing on, there's kind of a lot of overlap, but how much overlap is meaningful between different species? What can you really take away from this? And so I decided to do a different approach, actually, using Surface. It's an R package. And Surface is pretty cool because you can incorporate both morphological data and phylogenetic relationships to infer potential adaptive peaks that species are converging towards. And so this has been done with anolis lizards. This is kind of the most idealized version of what this would look like, where you see these different branches are colored based on these species being on their way towards these shared adaptive peaks. So pretty cool. Basically each of these Greater Antilles islands, you see anoles in this case that share a lot morphologically in common or are on their way towards it. With my stuff, I ran Surface, and the story is a little bit different. So that we don't see a bunch of branches of the same color. If we kind of zoom in on each of these parts of the phylogeny in each of the regions really quick, just to see if there's any similar colored branches, you'll notice, no, there is not. So all these different hypotheses don't really seem to be supported because there just isn't widespread convergent evolution going on with these frogs, which surprised me a lot because they kind of all seem to do similar things, ecologically. Morphologically, there wasn't some sort of cool story here. So again, I essentially refute all these different hypotheses, which is a little frustrating, but the story gets cooler, I promise, or at least I think so. The thing that I think this really suggests is that there might just be this many-to-one mapping phenomenon going on where there's a lot of different morphologies that could lead you to having a similar ecology. So maybe there's some sort of ecological convergence story going on here between the different species across these regions. But morphologically speaking, I don't see much. Now, again, I wanted to see if widespread morphological convergence can feed into community convergence. I don't have that first part, but could there still be some sort of cool community convergence story that's not predicated on morphological stuff? And again, I have a bunch of different hypotheses. I'm not going to run through them, except the first one, you remember I have these different landscape types, the dry forest, the lowland wet forest, and the upland, colder wet forest. You would expect that if those environments are super important for determining what types of species can be there, that there might be convergence in the species richness of those different environments. And so, yeah, again, I have all these other hypotheses. I'm not going to run through them because I don't have a lot of time. But I wanted to look at just strictly species richness in these different environments. So really quick, the way that I got data for my communities was through transects. This is kind of an idealized version of a transect, basically just counting all the frogs you see. Across all these different landscapes in those different regions of the world, we did 15 to 20 different transects, sampled day and night for over 400 of them. So it's been a pretty long, ongoing process. And we've resampled species richness for the analyses in like 20-meter subsets. And I looked at the direct developing frogs alone, and then also all the frogs together that were encountered on transect. I followed this kind of classic older method from Schluter and Rickliffs, where you just run ANOVAs with habitat type as a fixed effect and region as the random effect. And you can calculate the variance in species richness that's attributable to either habitat or region. And if you have cases where habitat is like more responsible for the variance, that suggests some sort of community convergence. Basically, these lowland places all seem to have relatively similar species richnesses, et cetera, et cetera. So what does it look like? It's all kind of distilled down into these couple of figures, but on the left you can see the direct developing frogs. Similar to that study with the finches from years ago, there's kind of this cool like relative pattern where species richness of the direct developing frogs kind of goes up from dry to warm wet to cold wet. You kind of see the same thing with all frog lineages combined, which is cool. But what's responsible for the variation? It actually seems that habitat is the most responsible for the variation in species richness when you compare these different places to one another. It's like the full model using all the regions, if you just compare Puerto Rico and Hispaniola. In most of these cases, the habitat component is much larger than the regional component, which suggests some convergence in species richness, both in an absolute way and in this kind of relative pattern that I've graphed. Same thing with all frogs combined, which is pretty cool too. And the regional component, again, is quite low in all these cases, except when you remove dry forest, which I'm happy to talk about later. But anyway, kind of a cool story. So again, I did not find a bunch of widespread evolutionary convergence with this system, which I was expecting to, but I do see some broad support for community level convergence in the direct developing frogs and in frog communities in general. Yeah, I talked about the many-to-one mapping. I think there could just be sort of something weird going on where it's like there's a certain set of ecological niches available. The species occupying them aren't similar, but there's still like a limited number of them, and thus the species richness is kind of consistent in these places. There's a lot of additional directions I want to go with this. I still have data I need to add in from Jamaica that I finished collecting this last week, and more morphological like convergence analyses I think I can do. I also want to add in the ecology to see if there is ecological convergence happening from that transect data. So a lot of cool things to come. And yeah, really quick, I just want to thank everybody from my lab, all the different, like the massive number of people that have helped me with these transects and things. It's been really an amazing experience. So thanks to all of them, and thanks to all of you for coming to my talk, and I probably have run out of time. Thanks.
Okay, hello everyone. I'm Malahatosada Dianat from Institute of Vertebrate Biology, Czech Academy of Science, Czech Republic. And I want to talk about DNA methylation as a marker and mediator in senescence. I will show you preliminary result about this topic. First of all, we need to know what is senescence and transmission. Gradual decline in the condition of individuals called senescence by aging, which is an ongoing, complex and inherent process. And the onset and rate of aging is different from individual to other individual. And the age of a parent affects the quality and survival of their offspring. It means that as parents get older, their ability to take care of their children diminishes. Still, we are at the beginning of understanding how parental aging affects their offspring, but to know this, we need to know the correlation between the level of senescence in the parents and the characters of their offspring. And also, we need to find the physiological changes in the offspring that comes from their parents. To do this study, transmission of aging from parents to offspring, I was using methylation as an epigenetic marker which is affected by changing in the environment, by pollution, by aging, or by stress. And methylation also is a marker that can transmit from parents to offspring. And what is methylation is adding a methyl group to cytosine. I cannot show, let me. Adding a methyl group to cytosine where when cytosine is next to guanine, and we call it CPG island. And adding this methylation, if it happens in the promoter of the gene, can change the regulation of gene expression. And it was shown that most of the genes that were affected by methylation became methylated was the genes that were responsible for development, differential, or also metabolic process. To study methylation as an epigenetic marker, I am using a model, Notobranchius furzeri, a killifish from the ephemeral savanna pools of east of Africa, and they have very short lifespan and they grow very fast, and they showed that they don't have any parental care. The aim of this study was to check the effect of aging on the amount of DNA methylation on somatic tissue. We chose FIN. On the parental individuals, also compare the amount of methylation between FIN and also gonads, and to see if there is any alteration by aging in the parents, if they can transmit to their offspring or not. For doing this study, I was using a method of next generation sequencing called DDRAT sequencing, double digest restriction site associated DNA sequencing, but it was a modification from this DDRAT sequencing. We call it EpiRAT. Actually, each sample goes for preparing library two ways. Once using EcoR1 and MSP, and once EcoR1 and HPA2. In DDRAT sequencing, we use just one pair of enzyme, but here in EpiRAT, we use 2 pairs of enzyme, and what is the difference? The difference is that MSP1 is an enzyme that is not sensitive to methylation. It means that it cuts the DNA and it doesn't care about the methylated site. However, HPA2 is an enzyme which is sensitive to methylation, and it doesn't cut the DNA if it is methylated. Then, when we have the library from these 2 pairs of enzyme and we sequence them, we compare these 2 libraries to find the methylated site. How was our experimental design? Of course, we needed parents and offspring. For parents, we had 6 male and 5 female from 3 different ages. We collected the FIN clips from age 1, 8 weeks, when they were 8 weeks, 22 weeks, and 30 weeks. I am showing here the sampling. It means that finally we had 18 samples of male and 15 samples of females parents. And from all these 3 ages, we also had offspring, and they had all 4 weeks. I am showing here the number of samples for each age of the offspring. And I should mention that apart from the FIN, we collected the gonads from age 3 of parents. When we had the sequences, first of all, we checked them for their quality. We filtered the bad quality reads. We mapped them with the reference genome, and we extract the read counts. We did the normalization and the rescaling. We filtered the insufficient representative of the loci, and we convert the data, the read counts, to binary data, and finally the differential methylation analysis was done. I didn't want to use all number of loci. I wanted just to work on the loci that are represented well across the samples. That's why I used the library coming from the enzyme which is not sensitive to methylation, means the one that I showed MSP with MSP. And to filter the loci, I checked different minimum number of reads per locus across samples, and I found that by increasing the minimum number of reads per locus across samples, the number of loci decreased. However, the proportion of methylation was stabilized here around 33. It means that I chose this 33 as a threshold to filter the loci, and the final number of loci after filtering was 3,485. Still, I did another filtering. Now I have the all loci here from the previous step. This time I am removing the loci that they don't show any differentiation or variation, this time based on the library coming from the enzyme which is sensitive to methylation. And the final number of loci I am showing here in the heat map was 926. You can see here the samples and the loci in the heat map, and here the gray part are loci that are methylated. And the dendrogram are showing, grouping the samples that have similar methylation pattern across the loci. Now I am showing the amount of methylation for parents, separately for female and male, through aging from age 1 to age 3 of the fin clips. And we found that generally the amount of methylation is increasing by aging for both female and male. But however, the amount of methylation shows relatively high individual variation. It means that if you look at, for example, this sample F1, the pattern of the amount of methylation is different from the other sample. The same for the samples of male. And when I compared the fin clips with gonads of the parents of male and female for the amount of methylation here, you can see that the amount of methylation in gonad is higher than fin for both female and male, and also the amount of methylation for testis was higher than the amount of methylation in ovary. As we had very well annotated reference genome of Notobranchius fusariae, I was interested in the loci that changed their methylation status, and I wanted to know what is the functional implication of these genes. It means that I wanted to know the loci that kept their status from age 1 to age 2. You can see here that I found 26 loci kept their methylation status, and when I checked these 26 loci for their functionality, I found that yes, most of them are responsible or involved in developmental process, differential process or metabolic process. And I am showing here the loci that were not methylated in age 1 and they became methylated in age 2. I found 3 loci. I forgot to mention that here I am showing examples just for the samples of female parents. And these 3 loci, when I checked them, I found that they didn't show any functionality, or I checked for the loci that they were methylated in age 1 and they became unmethylated in age 2, and I didn't find any loci. To summarize my talk, generally, the amount of DNA methylation is mainly affected, influenced by the age of the individual. It means that the methylation, the amount of methylation is affected by age of the individual, and they don't transmit to the offspring. Probably they reset in the zygote. And what is the next step? The next step is, as I mentioned, we have the reference genome of the Notobranchius fusariae. We are interested in the functionality of these genes. This time checking the male parents and also the samples, the changing of the loci status from age 2 to age 3, and also checking the methylation status in their offspring. Thanks for your time. I will be happy to answer your questions.
All right, So thank you all for being here. My name is Jocelyn. I'm a master's student at Université du Québec à Montréal, and I'm supervised by Professor Stephen Campbell and co-supervised by Jean-Baptiste Leduc from Université Laval. So today I'm here to present you part of my master's project, and I entitled my presentation, Pangenomics Reveals the Genetic Basis of Phylosphere Methalobacterium Adaptations to Host Species in a Temperate Forest. So what is the phylosphere? The phylosphere is all the aerial parts of the plants, notably the leaves, and it's an important microbial habitat because it sustains diverse bacterial communities, and those communities play important ecological roles in plant health and also in ecosystem functioning. And because every host has its own specific leaf morphology and leaf phytochemistry, the phylosphere sustains a diversity of niches for microbes. Methalobacterium is a ubiquitous bacterial genus of the phylosphere. This genus is part of the Methalobacteriaceae family, and it's composed of 104 species. And these bacteria are known for their plant growth promoting characteristics. So they synthesize phytohormones such as auxine and cytokinins, which stimulate the growth of their host. These bacteria are principally chemoheterotroph, but what's very interesting about them is that they are also facultative metallotroph. So that means that they can consume one carbon compound, such as methanol, that is secreted from the leaves during the tissue growth. Now, a lot of the strains can do aerobic anoxygenic photosynthesis, and that photoheterotrophy is very beneficial in low-carbon environments, such as the phylosphere. So Methalobacterium has been shown, the species composition of Methalobacterium, it has been shown that it varies with host species. But what we don't know yet is what are the genes that are responsible for the adaptation of Methalobacterium phylosphere population to different host plant species in the phylosphere. So for those of you who might not be that familiar with pangenome, a pangenome consists of the totality of the genes that are contained in all genomes of a given taxa. It can be a species or it can be a genus, as in my case. And the pangenome is composed of core genes and accessory genes. So core genes are genes that are found in all genomes of the taxa, and accessory genes are found in only a subset of genomes, or they can even be unique to a genome. And those accessory genes are often associated with secondary metabolism and also with the structure and biochemistry of the cell surface. Now, a question that is subject to much debate in pangenomics is, are accessory genes mostly adaptive, neutral, or deleterious? So this big question has led me to my own research question, which are, what are the genes that account for host species adaptation in Methalobacterium population that live in the temperate forest phylosphere, and what's the role of the accessory pangenome in those adaptations? My hypothesis is that accessory pangenome diversity is partly driven by adaptation, and therefore, accessory gene composition of Methalobacterium communities and population should vary with host tree species, and some genes should be associated with particular hosts. So here's a quick overview of our method. So we did two things in parallel. The first thing we did is collect genes from natural Methalobacterium populations, so collect metagenomes, and we constructed the pangenome of Methalobacterium. And then once we add those two elements, we mapped the metagenomic reads onto the pangenome and then proceeded to our statistical analysis. So for the metagenomes, we sampled in the summer of 2023 at the Laurentide Biological Station here in Quebec, and we collected 25 samples. We have 4 tree samples, so balsam fir, eastern white cedar, sugar maple, and American beech, and we have 1 shrub species that is a beaked hazelnut. And to collect the microbial cell, we washed the leaves with a phosphate buffer, and then we proceeded to the DNA extraction, and we did a metagenomic shotgun sequencing to get approximately 75 million of reads pair per sample. To construct the pangenome, we used the software Envio. So the first step was to select 1 reference genome per species. So we gathered 104 reference genome of Methalobacterium, and then we predicted the genes on those genomes, we functionally annotated those genes, and we finally clustered homologous genes based on their amino acid sequences similarity. And as I said, we did competitive mapping with Bowtie 2 to map the metagenomic reads onto the pangenome. And for the analysis, we assessed the relative abundance of genes using the coverage depth value, and we applied rarefaction for all analyses. So we found in all our reference genome that the pangenome of Metallobacterium had 70,695 genes in total, of which only 1.6% of the genes were core genes. And out of all the accessory genes, we found over 38,000 that are singletons, so genes that are found in only one genome. And out of all those genes, including core genes, we found 38,069 hypothetical proteins. So these proteins are, well, they're genes that are predicted to encode a protein, but we don't know anything about the function of those proteins. And these hypothetical proteins made up over 50%, well, 55% actually, of accessory genes. So in summary, we found a very high diversity of genes in the pangenome, and we found a lot of rare genes and a lot of unknown genes. Next, we investigate how accessory gene composition of Metallobacterium communities varied with host tree species. And we found that host tree species explain 25% of the variation in accessory gene composition of Metallobacterium communities. The figure we're looking at now is a distance-based redundancy analysis, and all the points are Metallobacterium communities, or samples, and the colors are for different host species, the triangles are for gymnosperms species, and the circles are for angiosperm species. And the communities are ordinated according to their accessory gene composition. Now, on the first axis explains almost 45% of the variation in accessory gene composition, and the second axis, 27.5%. And what is really interesting about how those communities are ordinated is when we look at the two, how the points are ordinated according to the two axes. So if we take axis 1, we've got all the host tree species on the left-hand side, and on the right-hand side, we've got the shrub species. And if we take the second axis, on the lower side, we've got all the two gymnosperm host species, and on the upper side, we've got the 3 angiosperm species. So we wanted to know if there were any genes that were associated with those particular type of host, so we did differential abundance analysis. And we found 64 genes that were associated with different type of host, and of those, there were 20 hypothetical proteins. So we found 3 genes associated with gymnosperms, 13 with angiosperms, 42 genes associated with trees, and 7 genes associated with shrubs. Now I put only a couple of examples, and I have still to dig deeper into what are those genes and what they're doing, but what struck us is that there are a lot of genes associated with trees that are involved in aerobic anoxygenic photosynthesis. And that kind of makes sense because there is more sunlight that reaches tree leaves than shrub leaves, so it's an advantage for bacteria if they can harvest that light. And also we found a lot of genes associated with metallotrophy and amino acid metabolism. Now we asked ourselves, does the host directly select the genes, or does it select taxa, which themselves come with their own set of genes? So in order to do that, we went from a community level analysis to a population level analysis. And here by population, we use a very technical definition. So we define a population as a bunch of bacterial cells from the same Metallobacterium species that is detected on a given tree. So now we're looking at another distance-based redundancy analysis, but here all the points are not communities, but populations, and all populations that are encircled in the same ellipse belongs to the same Metallobacterium species. So we found that accessory gene composition of Metallobacterium population is mainly influenced by Metallobacterium species identity. And it makes sense, and it suggests that Metallobacterium species are indeed coherent genomic unit. But we also found a significant interaction between Metallobacterium species and host species, and that means that there is an intraspecific variation in accessory gene composition that is explained by host species, and this is probably due to horizontal gene transfer between co-occurring populations. So next, we would like to investigate the link between bacterial gene and host phytochemistry and physiology, and to analyze the variation in single nucleotides and amino acids to detect concrete signals of evolution by host selection. So what I would like you to remember is that Methalobacterium accessory gene composition is shaped by host species, and that support the hypothesis that at least a portion of the accessory pangenome is adaptive. Methalobacterium species, as I said, are coherent genomic unit, but host species contribute to intraspecific variation in the genetic composition of population. And we found some genes related to photosynthesis, metallotrophy, and amino acid metabolism associated with either conifers or angiosperms or with trees or shrubs. So with that, I would like to thank you very much for listening, and I would like to thank everyone at the Campbell Lab for supporting me during my research. If you have any question, I will be glad to answer. Thanks. Yes.
That was really cool. I guess I have a lot of questions, but do you see any of these, like any linkage between the accessory genes and?
I haven't looked at that yet. This is maybe if I have the time to look into that, I will, but I haven't looked at it yet. So yeah, but it's an interesting thing to do. Thanks.
And like, do you see any I guess at a single nucleotide polymorphism level, or any, I don't know, something that you're gonna do?
Yeah, this is, yeah, as I said, this is like pretty much the next thing I'm going to look at, because I have the data, I just haven't had the time to analyze it, but it will be very interesting to, because now I've shown genes that are associated, but yeah, now I want to look more precisely at nucleotides. Yeah. Well, thank you very much, and have a good lunch.